Hi, I'm Judy Cole, the Executive Vice President and CEO of the MIT Alumni Association, and I'm delighted to welcome you to this web production of the MIT Alumni Association.
Please welcome to the stage Noubar Afeyan.
All right, well, hi everyone. I'm Noubar Afeyan. I did a PhD here in 1987 at a time when biological engineering didn't exist. And so I ended up doing a degree in the chemical engineering department. At the time, the really beginnings of trying to make engineers familiar with biology. And 30 years later, it's a great pleasure to share the stage with some of the faculty that you've seen here and hear about MIT at its best. I want to say a couple of introductory words, and then we're going to take questions. And I will implore you, if you're interested to pose questions, to use this website, P-O-L-L-E-V, pollev.com/MITQ. Some of you have already provided questions. Feel free to add them as we go. But just personally, I'd like to really say a couple of words about the context for all of this. We're here today in some way celebrating MIT's pioneering effort in biological engineering. Biological engineering as a terminology didn't exist about 25 years ago. And 20 years ago, thanks to really the leadership of Professor Doug Laufenberger and many of his colleagues, MIT tried to create a new discipline. And as such things go, there were a lot of naysayers in the beginning, both internal and external. The world didn't need yet another discipline. Mechanical, chemical, civil, electrical were plenty. And sitting here today, seeing the vision play out in the form of really things that I think just people within the previous disciplines may not have dared to do or interacted together to be able to do, is proof that that vision is being paid off. I've been a beneficiary of this, having participated with the department for some 18 years as a visiting committee member, and have had a daughter graduate just a couple of years ago with a bachelor's in biological engineering department. And so I'm here partly to, I think, really bear testimony to what they're doing. And I really would urge all of you, as I look around the room, who are alumni back here celebrating your own departments, to also take note of this brand new engineering discipline. It's not very often, once maybe every 50 years, is a new discipline born in engineering. And I'm very proud as an MIT graduate to be at the institution that really created this department. So much for that. I want to just kind of maybe start on that note and ask each of you, or whoever wants to comment, to just talk about what biological engineering is doing to students. In other words, what is it that we're educating? What is the output of this department, and where do you see them ending up? Maybe, Linda, you can start.
I'll start because I led development of the undergraduate curriculum, which was MIT's first new major in almost 40 years. When it launched in 2005, we just had our 10th senior dinner last week in graduation. And it really happened because every time there's a revolution in basic science, there needs to be engineering to translate that from the basic science into practical things people use through the lens of analysis, design, and synthesis. How do you analyze complexity in biology and make sense of it, understand information flow to create a set of design principles? And that's really, really the key. You have to understand the fundamental molecular science in order to build mathematical models of how it works that let you then make blueprints. So we're really teaching them the tools to understand the biology and then to design and build things out of it. And this has met with tremendous enthusiasm from industry, from other graduate schools. We get tremendous interest in our students. They're really learning how to understand biology in an engineering way from molecular systems to build things.
So one thing I'd like to comment on is that in our department, we also try to really foster not just studying in the classroom, but also studying outside the classroom. And so we do this through a variety of processes. We have specific classes that are focused on, for example, having the students come up with their own research projects. So they go, Linda mentioned this notion of design process. So they actually come up, these are undergrads that don't necessarily have a whole lot of experience, and they come up with these crazy ideas about projects that they want to do in biological engineering. And we step them through the semester going from kind of these vague, crazy ideas to things that actually are built, implemented, and can be tested and have real function in biological systems. So we incorporate that both in terms of the courses that we offer to the undergrads, as well as extracurricular activities. One that I like in particular is something called iGEM, which is this international genetic engineering machine competition where we assemble a group of 12 or 14 undergrads, many of them from biological engineering, and just tell them, go for it. You know, come up with some crazy idea that you want to be able to engineer in a biological engineering setting, and we're going to help you through the process and actually make it happen.
And I'll just add one thing. Last year, they did a crazy thing. Estrogen and progesterone sensors in endometriosis cells. I thought it was crazy. But it worked. We're publishing a paper, and now Ron's daughter is actually working in our lab this summer to take it across the finish line and translate it to Vanderbilt. It's just, I can't even believe that happened, and it did.
So just to move on and tie it back to some of the talks, I know many of you and many of the questions I'm getting have to do with the effect of some of these technologies in use with humans that may be posing safety risks. Whether it's viruses that are meant to detect and activate in certain cell types, deciding to do it in other cell types, whether it's editing while having a positive effect, also causing other unintended mutations, and likewise, Linda and yours. So maybe you can talk, as you know, as an engineering discipline, I always think that engineers try to do useful things before all the knowledge is available, and advance the knowledge and the usefulness at the same time. And so we don't really know what some of these things might do. Maybe you can each comment about how you think about this balance and how to deal with the unknown risks of some of these in human application. Ron?
Yeah, I mean, I think that's a terrific point, and it's, you know, we kind of view this as you mentioned, as an iterative process where we, one of the really cool things about biology and biological engineering is that it incorporates this iteration of science and engineering. So for example, in comparison to computer science, when I was mostly working on programming computers, I wasn't doing any kind of fundamental physics in the process. I wasn't trying to invent any new physics or gain insight to that. I just was doing the engineering of the computer systems. And in biological engineering, what you do is you have some hypothesis about how the system works, and then you try to modulate it. And in the process of modulating it, you end up learning a lot more about the system, which then allows you to then do better modulations, would then allow you to do, you know, have better understanding and so on. So I think this iterative process is really the way to go, and that's the thing that allows us to do things like have more predictable outcomes, more predictable systems. And so the way we approach this is by learning as much as we can about the systems, and then trying to engineer mechanisms that work well in a biological setting. So for example, I mentioned all these notions about looking at not just an individual biomarker to be able to distinguish one cell type versus another one, but let's look at many different biomarkers. Let's integrate genetic circuits that have a lot of redundancy in them. So really try to understand, how does biology work? Why is biology so effective even though it faces all these constraints and of gene expression noise and unreliability, and try to then incorporate that into the way we think about a new type of engineering.
Feng, you want to comment vis-a-vis the editing approaches?
Sure. So the technology that I told you about is about going into the genome of human cells and then making changes to get therapeutic effect. But what if that system not only makes the intended change, but also makes other changes? And so that's one of the major concerns that we have about advancing the technology toward therapeutic uses. And to do that, we have to also concurrently develop technologies that help us assess the specificity of the system. And so in addition to developing the technology itself, we also have to work on and thinking about what's the auxiliary technology, the supportive technology that sort of go around this main system so that we can that can help us understand it more, and also to be able to continue to iterate and optimize the basic system. Other things besides the specificity question, the enzyme is a bacterial enzyme, so it's something that's foreign to the human body. When we put it into a patient, we're exposing it to the human immune system. What type of immune response, what kind of inflammation this foreign protein may cause, that's a second area of major question that we have. So there are a number of these different questions that we think about, and we try to develop new systems to help us understand and to be able to iterate and make it better. But more importantly, as we move forward, we have to be very thoughtful, and the key is to be careful and be sort of gradual in the way that we advance it and apply it, looking at cost and risk, or risk and benefit of the given technology as we apply it is also very important. We don't want to prematurely apply it to any system where we don't have a good understanding of what the outcome will be, and also what the potential risks might be. And we really need to know all those things in order to mitigate the risk and also to responsibly develop the system.
So in my case, I'm kind of the end of the food chain. I work a lot with preclinical safety and toxicology in the pharma industry, and they're already using our livers and our other models to look at how drugs will affect humans instead of mice, and we're learning some very interesting things. What is critical is to build more and more complex systems. You may have heard human body on a chip. We're actually building a physiome on a chip. A physiome is an integrated physiology where we actually understand how we designed it to get specific information. So all the kind of technologies that you heard about here, we're hoping to be able to test in our in vitro systems, because we are bringing even the adaptive immune system we're starting to develop. We have a project with a pharma company. So we're building really complex interacting organ systems. We just finished a project with DARPA, 10 interacting organ systems, including a model of a brain from Rudolf Janish's lab, and others, 10 interacting organs for a month, so that we could really start to look at crosstalk between these. And we're really at early stages, but these all integrate so that we can build the test systems thoughtfully designed to understand how gene editing would work in our endometriosis cells, for example.
So eventually, Linda, you're saying that you're going to create in various ways, artificial life that can then house the artificial intelligence that other departments are working on at MIT. Is that basically
you weren't supposed to say anything about that.
Okay,
That's
all right,
secret.
we can't say that. All right, we didn't say that. For those of you online, just forget it. Linda, maybe just for you to quickly comment, and I'm mindful of time, just about the environment, the budget environment. We've heard a lot about potential cuts in NIH funding and kind of an outright assault on facts. But generally, can you talk about how you think the next few years may play out and what MIT is doing about it?
So I'm on the advisory council to the director of NIH, Francis Collins, and we actually just met Thursday and Friday this week around a very controversial proposal NIH was making to cap the number of R01 or grants that any single investigator could have, and there would be 43 people at MIT who would be caught in that web. And our committee, assisted by some wonderful folks at Sloan, convinced the NIH leadership that this was not as probably productive as they thought. NIH, Congress has proposed a $3 billion increase in their budget. And so right now, the budget process is not finished. There may end up being a continuing resolution, et cetera. But NIH, at least, has very, very good relationships with Congress and with Trump. So Francis Collins just met with Trump with a bunch of pharma. They're making public-private partnerships. So I'm not as pessimistic as you might read in the press. NIH is tremendously trying to be a good steward of taxpayer dollars, but also working very, very hard with Congress and with Trump and his team to have, at least for biomedical research, the need to keep not only the current levels of funding, but perhaps to even increase these levels of funding to address the challenges we have. As for other areas, I'm not quite as familiar because I've been completely buried in helping NIH navigate this issue.
Great. So let's see, all of us who've done PhDs here have taken qualifying exams, including you, Ron. And there's a question of this list that I want to pick up on to put Ron on the spot. So the question reads, can it be shown that the cellular environment, including all genetic materials, proteins, etc., is Turing equivalent and capable of universal computation? Thank you for that question.
Okay.
And thanks that it's not my exam.
Sure.
Ron?
Yes. So actually, the way you accomplish this is by showing that you can build logic functions such as not or. So if you're able to build not or Boolean logic functions using DNA and interactions between proteins and DNA, and then you can show that you can connect these NOR gates with one another, that essentially gives you universal computing. And so we've been able to show NOR gates. The other way you can do it is by building not AND or NAND gates as well. So either NOR gates, NAND gates, you can connect them up and build anything that you want, essentially.
Perfect. We're running out of time. There's a lot of questions we're not going to get to, but I'm assured by the Alumni Association organizers that these questions will get to the panelists so that they may be able to answer, or outside during the break you can get some answers. I'm going to be back for another such panel, and we'll continue your line of questioning then, but please join me in thanking the panelists. Thank you.
Thanks again for joining us. For more information on future MIT Alumni Association productions, please visit our website.